Hello everyone, today I'll be presenting my poster over sodium potassium beta subunit knockdown in Aedes aegypti mosquitoes. As we all know, mosquitoes are vectors for deadly diseases, specifically Aedes aegypti, who carry yellow fever, West Nile, Zika, and dengue. Mosquito pesticide resistance is becoming a major problem as mosquito populations continue to rise, so methods to control these populations are becoming a priority within research. A nutrient-sensing pathway is an ideal target as female mosquitoes will uptake blood for amino acids to produce yolk proteins for the development of their eggs. Sodium potassium ATPase was found to possibly interact with cat amino acid transporter CAT3, specifically the beta subunit of sodium potassium ATPase. This was discovered by using a yeast 2 hybrid assay and having the CAT3 as our bait protein. Sodium potassium ATPase is an ion transporter consisting of two subunits, the alpha and the beta subunit. This protein is found in the cellular membrane and is thought to interact with nutrient sensing pathway, specifically our CAT3 protein. Sodium potassium ATPase beta subunit is responsible for making sure that the alpha subunit of the ATPase is folded properly and directed to the cellular membrane. CAT3 is part of a nutrient-sensing pathway that signals for yolk protein precursors to be transcribed. Our aim was to determine if the knockdown of sodium potassium ATPase beta subunit would have physiological effects on mosquitoes, specifically Aedes aegypti mosquitoes, by analyzing the number of eggs they laid and the hatch rates of these eggs. We hypothesized with the knockdown of the sodium potassium ATPase subunit beta gene would lead to less cationic amino acid transport and in turn lower the production of yolk proteins. We predicted that the disruption of the yolk protein production would lead to smaller number of eggs laid and lower hatch rates. So for our methods, we first designed our primer, which was going to be used to elongate our gene of interest. We then extracted RNA from mosquitoes to have in order to reverse transcribe this into DNA. And this is where our cDNA synthesis technique was used. We then used our DNA with our primer to amplify our gene of interest, and we then place our gene of interest into a topo4 plasmid where it was cloned and transformed then into bacteria. And this was done in order to colonize and amplify our gene of interest and have a stock for later use if need be. We then re-isolated our plasmid from bacteria and we amplified out our gene of interest in order to have a template for our dsRNA synthesis. And our dsRNA synthesis was performed by using a T7 RNA polymerase. And within our gene of interest, there was a region where this T7 RNA polymerase could bind to. And for our dsRNA injection, we specifically use 1 microgram per microliter of dsRNA, and it was injected directly into the thorax. And over a 72-hour period, every 24 hours, we measured the mortality rates of our mosquitoes just to determine if this had any physiological effect on them. So for our results, as you can see in figure 1, this is our 72-hour period for our mortality rates and our survivalship. And as you can see from the 24-hour period mark, we have roughly 83% of our injected sodium potassium ATPase mosquitoes still alive, while our GFP injected mosquitoes are around 91% still alive. And if we move to our 48-hour period, we can see that 78% roughly of our sodium potassium ATPase injected mosquitoes are still alive, while 89% of our GFP injected mosquitoes are still alive. And finally, at our 72-hour period, we can see that 76% of our sodium potassium injected mosquitoes were still alive compared to our around 85% of GFP injected mosquitoes. So roughly a 10% difference between the two injections. So if we move to figure 3, we can see the egg number lay counts that we observed from our two groups. And we see that GFP injected mosquitoes have a higher number of egg counts laid compared to our sodium potassium injected mosquitoes. And Because we don't see a broad standard deviation, we concluded that this was a significant difference. And if we move down now to our figure 3, we measured our hatch rate percentage between the two GFP and sodium potassium injected groups. And we can see that because we have such a broad range for our standard deviation, we could not conclude that there was a significant difference between the two. And so in conclusion, the significant difference in egg lay count shows promising results in our sodium potassium ATPase knockdown. As the yolk protein production could play a factor in the decreased number, but confirmation of this will need to be determined through qPCR analysis of vitellogenin expression. And overall, additional studies need to be performed to confirm our sodium potassium ATPase knockdown, both egg lay counts and hatch rate data, so that the targeting of sodium potassium ATPase beta subunit could be a means of possible targeting for novel pesticide production. And so I'd like to thank Dr. Imo Hansen, Dr. Matthew Pinch, Yashuda Kendall, and the New Mexico Alliance for Non-Minority Participation staff for allowing me to conduct this research and present it to you all. Thank you.